Hello everyone, I'm Rubaiat Ahmed from the Laboratory of Population Genetics, Department of Biochemistry and Molecular Biology, University of Dhaka. The topic of my flash talk is most frequently harbored missense variants of human hACE2 across different populations exhibit varying patterns of binding interaction with spike glycoproteins of emerging SARS-CoV-2 of different lineages. This study has already been published from our lab, and today I will be discussing a little bit of the background, the methodologies, and the results of our research. To start things off, we all know about the SARS-CoV-2, which has spread all over the world, causing the COVID-19 pandemic. Till date, there have been more than 691 million confirmed cases, with the death toll exceeding 6.8 million. Despite mankind's best efforts, the virus continues to circulate the globe. Since its inception in late 2019, the virus has given rise to many variants like Alpha, Mu, Delta, Omicrons, and many more that differ markedly in infectivity and transmissibility. SARS-CoV-2 causes respiratory tract infection, and the receptor binding domain of the viral spike glycoprotein binds to the host receptor ACE2, and the virus is internalized. Mutations of the RBD and the polymorphisms of the ACE2 receptor alter the binding and may affect infectivity. As we can see, not all regions of the world suffered equally from COVID-19. So, the host genetics that differs among ethnicities might play an important role. Therefore, the objectives of our study were to evaluate the roles of the spike protein RBD mutations in binding to the ACE2 receptor, and we also wanted to explore the role of ACE2 polymorphisms in population-specific susceptibility to different variants. To study this, we mined public databases for mutations in the RBD and polymorphisms of ACE2 in different populations. Then we created protein models and performed molecular docking between the model RBDs and ACE2. Later, we did MMGB on the complexes and performed molecular dynamic simulation on selected complexes again. Now, coming to the results. We obtained the most prevalent missense variants of ACE2 within the African, Latin American, non-Finnish European, and South Asian populations, and we created models for Delta, Mu, and 4 Omicron subvariant RBDs. Looking at the molecular docking score, we observed substantial differences among the binding of different RBD variants with different ACE2 models. Omicron BA.1 and BA.2 have the highest docking scores with fairly all the ACE2 variants. When we wanted to have a closer look at the molecular level interactions, we also saw differences in numbers and types of bonds that are present between different complexes. For instance, Omicron BA.4 had high number of hydrogen bonds with reference and Latin variant ACE2, but it reduced in the African variant ACE2. The difference between the binding interaction is also seen in the 3D perspective. The binding pattern changes for the mutated residues of both the RBD and polymorphic ACE2 residues, and some bonds are diminished in the mutant proteins, while some new bonds are also formed. The bond length are also affected by the mutations. Comparison of binding energy among different RBD variants with ACE2 variants reveal differences in different energy parameters, which contributes to the overall scores. The difference in binding patterns are reflected in these results as well. Lastly, molecular dynamic simulation revealed the stability of different complexes between Omicron BA.1, BA.2, Delta, and Mu with different ACE2 variants by RMSD and RMSA values. Omicron BA.2 had a lower average RMSD compared to BA.1, reflecting real-life scenarios of greater spread of BA.2. The Delta variant had also more stable binding with South Asian ACE2 variants compared to the wild type. So, what did we understand from the results? Both RBD mutations and ACE2 polymorphisms alter binding patterns, which is demonstrated by all of the available parameters. BA.2 binds more stably compared to Omicron BA.1, causing greater spread, and this study might shed light into real-life transmission rates. To know more about this study, please visit my poster at the virtual poster hall. Thank you.